keep it waiting room. Very good morning to all my participants in the name of Almighty Allah. We are today starting our session. As usual, probably it will be sixth session today on this Saturday. The speaker of the day has already joined us. Dr. Sham Zawar is already with us. Most welcome, sir. Most welcome in this morning in our this online program, sir. I'm really humbled to see you over here.
Thank you.
And it's my pleasure, sir. We are really obliged that you have accepted our invitation and you have spared your time despite your busy schedule, and we realize your busy schedules there. But I'm really grateful to you, sir.
Thank you. Thank you.
You have accepted our invitation and you have been with us today, this morning. My dear colleagues, this session will be probably a very interesting session. Sir, my participants over here are internees. They are mostly the internees and postgraduate students. You know, we have already discussed that our motto and that objective of this program is that we will see our students not looking after the public or government jobs. Rather, they can see their future in private sector also, and they can be job providers rather than job seekers, as you have been the best example for them. Now, my dear participants, we are really lucky and fortunate to have this morning with us Dr. Sham Zawar, who is basically a veterinarian. He did his BVSc from Mumbai Veterinary College way back in 1968-74. Then he did his FVSc and then PhD in animal production. And then he has done his additional courses in management, 2 years management course from IIM Calcutta. Then he also did his advanced managemental courses from London Business School and Harvard Business School. So this is a brief about his, academic qualifications. A highly learned person with us this morning. He has a vast experience of 47 years in assisted reproductive technologies. A man of, I can say, high caliber in embryo transfer technology. Rather, the first man who has done his who has published his, the research and his this thesis, PhD thesis in super ovulation and embryo transfer in case of cattle, sheep, goat, and the first veterinarian probably in India who has started the embryo transfer technology in case of sheep and goat. Then he's a man with so many, the versatile, the so many, memberships of high reputed those committees and the societies. He has been a guest speaker representing the continent of Asia during 23rd World Veterinary Conference at Montreal, Canada. And he has also been, you can say, the international sheep and goat expert to prepare a project report for government of Nigeria. He is also a member of Health and Safety Advisory Committee, USA. Then member of Central Advisory Committee that's established by the Ministry of Animal Husbandry and Dairying. Also a member of Advisory Committee constituted by the Planning Commission of Government of India. He has been awarded by His Excellency President of India, Dr. ABJ Abdul Kalam, for his outstanding work in rural development. So he has been awarded for that. So now presently, if I can say, presently he is associated with JK Trust. JK Trust is basically an NGO that's operating two state-of-art embryo transfer and in vitro fertilization labs in Chhattisgarh and Maharashtra. That has been that JK Trust has been founded way back in 1945, driven by the ideas of its chairperson, that's chairman of that Raymond Limited, Mr. Gautam Hari Singhania. And this society is committed to augment the quality of life of people of rural areas. Now, very recently, just few days before, and I received that letter from the Dr. Sham Zawar. He has been appointed as a national mentor for assisting IVF labs under a Rashtriya Gokul mission by Ministry of Fisheries, Animal Husbandry and Dairying, Government of India. Congratulations, sir. You have been appointed as a national mentor. I do humbly salute you and congratulate you for receiving this position in the Government of India, and you'll be the man guiding the universities, many other organizations, whether in public or private sector, to establish IVF and ETT labs. So most welcome to this webinar this morning. My dear participants, this was a brief about, the speaker of the day. I believe this session will be very, very interesting. It's now, sir, we have also been joined by our those faculty members. Our dean couldn't make it this morning with us because of his poor health. Otherwise, he was very much interested to be there with us, but because of his some health issues, he's not able to join us this morning. We have with us Dr. Noor Alam Tufani. He is actually in charge of our clinical complex and in charge of these internees, is a course director also. We have with us Dr. Puttu, many other dignitaries, many other faculties besides our students. Most welcome, sir. Over to you, sir.
Thank you very much for such a wonderful introduction you gave about me. And just to correct a small correction is that I retired from JK Trust. As I told you, JK Trust Raymond, after putting 47 years of service just on 31st of October, and on 1st of November, I took this assignment from Government of India as they have appointed me as a national mentor. So I'll be visiting all the 30 labs which are located in different parts of the country. Unfortunately, I saw there is no lab in the Kashmir, and we should have one lab over there also. So if you send a proposal to Government of India, I'll also personally follow up. And why not? Almost every state has got a lab. So except some Northeast, they don't have. But Kashmir, we can have one lab over there. So I'll try for that. Thank
Yes, sir, for that, actually, we have a division of edible biotechnology over here. So they are looking after that embryo transfer technologies, and it will be my pleasure to share with you that the Nuri, the first, clone in case of Pashmina goat that has been developed by this institute in our, the lab there at Shohama Alasting in the animal biotechnology division. So many scientists are there involved in that, but as such with the name of ETT lab, probably it's not existing there in, but this is a very good offer from you. We will see that our reproduction division, animal reproduction division, just takes some leads in that, and you'll be there with us. Thank you for that, sir. Thank you.
Yeah, because there are some veterinary universities in the country. They also have got a funds received from the ministry to establish the ETT IVF labs. Like they have one in Patna, they have in Andhra, a couple of universities. They have one in Maharashtra. So why not try at in your state also?
Sure, sure, sir. Sure, sir.
I'm always amazed. I've been visiting Kashmir many times, right from 1975, and I had been going on a horseback to see on your alpine ranges the sheep in initial part of my career. That was well back in 1975, and I remember one doctor, Wani.
Yes, yes, sir. Yes, sir.
Who was there, and I used to interact with him a lot.
He is no more now.
Yeah, I remember. Yeah.
Yes, he was director that time, sheep husbandry department.
Right.
Yes, yes, sir.
Okay. So how do you want me to proceed? Tell me.
Anything we want you to share your experiences and your those, the work the NGO is doing and your those labs,
No,
IVF labs, mobile labs.
Do you want me to quickly go put my presentation and
Anything, sir? It's up to you. Now we have handed over our whole session to you. Now you just arrange it as per your.
No, and I will love to have many more questions, but let me go through quickly through my presentation.
Okay, sir.
For you
can
people
share
to have an idea, for which I think you will have to make me host.
You can share the screen, sir. No issue, sir. You just share the screen.
Okay, I'll do that. 1 minute and let me see. Just give me a few minutes. 1 minute.
Sure, sir. Sure, sir. It's just coming there.
Yeah, I have to just see locate.
Yeah, yes, you just see your presentation, please, there.
Yeah. Oh, this is something different.
This is what we have. Yes.
Very nice. I liked it. Yeah. Oh, no. I didn't locate it and kept it.
No issue. No issue. Let it take some time.
Yeah. Go to my. Just 1 second. Huh? 1 second.
Sure, sir. Sure, sir. No issue at all.
वीडियो बंद कर दो, वीडियो बंद कर दो. Just give me 1 minute, huh?
Sir, no issue. Take your time, sir.
Yeah. Desktop presentation. वेट बोल कर शेयर कर दे. Is it
There,
visible?
sir.
Is it visible?
Yeah. Sure.
वीडियो चल.
Good, sir.
Thank you. Yeah. Yeah. Actually, right now, I'm not the CEO, but that's a little older presentation. And very recently, I did it for the veterinarians only. So, are you able to hear me clearly?
Yes, sir.
Okay, so I'll go through this. This is especially I'm talking about application of IVF technology in cows and buffaloes at the doorstep of the farmers in India. And this is my presentation outline. I don't want to go through that outline, and so that we can directly go on the presentation itself. You know, as I think you rightly said, I had a long journey. Normally, you don't spend so many years in one single organization, but I was fortunate enough to be there with the Raymond for almost 47 years. And we started with in 1970, established a sheep breeding farm in Dhulia, where we imported Australian Merinos and the Polworth from Australia. And we started working on the breeding of the sheep. Mainly the purpose was to improve the quality and quantity of the wool, for which we brought some sheep from Rajasthan, a breed known as Chokla. And because of that sheep breeding work, I used to have a lot of interaction with the government of Jammu and Kashmir in view of the sheep, because we have known for the Kashmiri Merinos. So with that, I joined them in 1975, and where we started working on embryo transfer in sheep. And that time, the sheep, everything was surgical. You know, the recovery of the eggs, the fertilized embryos, that was done surgically. Transfer was done surgically. And nowadays, it's all done through the laparoscopy. So even in 1977, when I saw the embryo transfer in cattle, even that also was done surgically that time. Even embryos were taken out surgically, even for the cows also, and transferred surgically. Well, that is become history now. In 1983, Raymond, everybody knows about Raymond. I did not tell the introduction of Raymond. So Raymond established a cattle embryo research center. Having an experience of doing embryo transfer in sheep, we thought, now let's work on the cattle. And we started working on various aspects of assisted reproductive technologies in cattle. And it was from 1983 till the year 2000, almost for 17 years, I worked on MOET, as that is multiple ovulation and embryo transfer, wherein embryos are made inside the uterus and then you flush them out. So we call them it as a in vivo. Then from 2000 to almost 2016, there was a clear-cut gap about my work in embryo transfer is concerned, because at that stage, we, JKTS, started operating a huge number of artificial insemination centers across the country. And we were operating in 10 states. Almost about, we have touched to the figure of almost 5,000 centers and covering near about 60,000 villages in the country, and benefiting millions of farmers. And we did millions of AI. I must say, because we are addressing to many of you from Kashmir, we made a couple of trips to Kashmir. I had wonderful meetings with your ex-Chief Minister Ghulam Nabi Azad, and also the next Chief Minister also, and many of your bureaucrats also. But somehow, it was almost coming to the final stage and it couldn't get materialized. But I think now my next CEO, to whom I have handed over the charge, Recently, he was in, I think he was in Jammu or I think in Srinagar, and he had good meetings over there, and they are looking for doing some work on artificial insemination and other aspects in your state. Okay, that's the one thing. But in 1997, this JK Trust was launched, wherein we were doing this AI program. But my interest in the embryo transfer was there already, and I was very keen to start some work on IVF. So in 2016, there's only 5 years back, we had set up a very high-tech laboratory near Pune, and all of, I mean, I can invite you on behalf of the JKTS, even though I'm not there now, but it's only a week since I'm not there. But any one of you interested can write to our new CEO, and I'm sure you can visit the lab. It's a world-class lab. You will see it. And in 2016, a unique thing is that we launched even mobile ET and IVF labs. So initial focus, when the JK Trust was doing AI program, and they are still doing artificial insemination program on a large scale in the country, the main objective was to reduce the malnutrition and create employment and poverty alleviation. I hope everybody is able to hear me clearly.
Yes, sir. Yes, sir. Yes, sir. Yes, yes, yes.
Okay. So we were selecting a educated unemployed youth from the village and giving him an intensive training to do AI. That's how we had a huge coverage of this AI program. Our present chairman is Gautam Singhania, the chairman of JK Trust. Rather, I have to address it that JK Trust because this is the presentation made when I was in JK Trust. So he is the chairman of the JK Trust and who is also chairman of the Raymond. Now, the JK Trust, they launched another division known as JK Bova Genix in the year 2016, mainly with the objective of this, starting the IVF and ET services, and not only restricting our work to the lab, but taking it to the field level and doing the services at the doorstep of the farmer. This is one of the flagship program of the government of India known as Rashtriya Gokul Mission. And under that Rashtriya Gokul Mission only, there are about 30 labs which are being provided funds from the central government. And that's what I was telling, why not we should have one in your state also. So even next week itself, I'm going to be in Delhi, so I'll take up this matter personally. And meanwhile, you people can send a project proposal for your university as well. Now, let's look at the different ways of the, I just want to quickly check as to in the total attendees who are here, around I've seen almost about for 50%. I mean,
50.
are there any postgraduate students in animal reproduction also?
Probably there are, sir.
Okay.
There are.
And faculty, there will be some faculty members also?
Yes, so faculty members are also there.
Great. Okay. And these are final year students?
These are internees rather.
Okay, they have already finished their course work, right?
Yeah, yes.
Okay. And they are now looking for their opportunities in future.
Exactly. That's why we are here.
Yeah. Whether they want to do masters or they want to start their own something,
exactly. Entrepreneurship.
And I request all of them, all the students, to take a clues from this. It's always better to be a self-employed than getting employed for someone. And I'm sure there are a lot of scope is there. If you go into the detail, you can just take small things out of it and try to do something in that. That's not a very big issue. And it's question of practice only. More the practice you do, I'm sure you will be able to master the technique. Nobody is born with a master skills. So one has to do the practice and one has to have that willpower also, that is very, very important. Without willpower, you can't do anything. Even at this age of mine, which is nothing, I mean, in India, we have become old, but that's not a correct way. Even though I'm 71, 70, I still do. I love to do embryo transfer myself. I love to do OPO. So if you have a willpower, you can do anything, age doesn't come in the way. So look at these different ways of breeding. One is the natural breeding, everyone knows. Another is the artificial insemination. Another, that's also ART. And the third one is this multiple ovulation and embryo transfer, which I talked some time back as a in vivo embryo production, wherein you select the donors, which are good animals, and you want to multiply them faster. You can produce the embryos inside the uterus, flush them away, flush them from the donor at when they are 7 days old, and then you transfer them into the recipient cows. So coming back to the sheep, our purpose in case of embryo transfer in sheep is to have a better quality of wool and increase the quantity and quality of wool by having a good genetics. That's the main purpose in case of sheep. In case of cattle, it is like for in our context, improving the milk yield. So the objectives are different, but the technology more or less remains the same. So this third technology, this multiple ovulation and embryo transfer, this was followed all over the world for quite a few years. And I was extensive traveling all over the world, I know how this transformation has occurred from the multiple ovulation and embryo transfer to IVF. And you will know once, having worked for 20 years into the MOET, I don't like even doing now any multiple ovulation and collection of embryos. Feel like doing everything outside the womb, having the artificial womb created into the lab. The uterus, almost we are providing all the environment in IVF, which I will tell you in more detail. So in case of MOET, as I told you, the whole purpose of this whole ART, whether you take MOET or IVF, is to multiply genetically superior animal at a very fast rate. A cow will give, or buffalo will give one calf in a year if she is a regular breeder. So you won't expect more than 8 to 10 calves from one cow or a buffalo in her lifetime. But say if you have got a herd of 50 animals and we have got one outstanding animal in that, which is much, much higher in milk yield and genetics, the best ways to multiply them is you have to use this latest technology of the embryo transfer. And because as I want to tell you, we have been using the males to the fullest potential. The bulls, we collect the semen and we dilute them, and from a bull, we make 100,000 doses in his lifetime. Whereas female, we are not utilizing the fullest potential. Even though she has got that potential, once a female calf is born, for your information, she has got theoretically a capacity to produce, she can produce about 60, she produces almost 65,000 oocytes, eggs. Oocytes are, it's like a female gamete from her ovaries in her lifetime. But we are only able to get 8 to 10 of those oocytes getting ultimately fertilized and resulting into embryo and pregnancy, because we were not harvesting those oocytes. And there is a technology now through IVF, you can harvest those oocytes at a regular interval. And I will show you an example of some of our elite cows, from which in 1 year time, we have established almost about 100 IVF pregnancies. That is really how the impact of IVF is seen. There is another cow from which we have touched about almost more than 125 pregnancies in, I think, 18 to 24 months. So you have been regularly taking out the oocytes, and I'll tell you the process later. But MOET, I'm closing it here because it has got a limitation. You can only do 4 or 5 times in a year, and you can't regularly do IVF, like IVF. Now, the latest technique, what we call ovum pickup, some people call it as aspiration, is nothing but production of embryos in vitro. Earlier one was a in vivo, and this is in vitro, means you produce the embryos outside the uterus. And in both the places, you are doing embryo transfer. So embryo transfer is common. How do you produce embryo is more important. One is inside the uterus and another is outside the uterus. This is very advanced breeding technique, definitely costly, but still it is worth. And it is exactly similar like test tube baby in human beings. No difference whatsoever. Almost majority of the equipments which are being used are the same equipment which you will find into the IVF human labs. Now, in case of this IVF, we can utilize the male and female genetics potential to the maximum extent. And like any MOET, when you are flushing the embryos, you are actually giving a massage to the uterus. Whereas in case of IVF, we don't touch uterus at all. It's only ovaries. Our target is to look at the ovaries and get the oocytes from the ovaries. Now, I don't want to go into the details of the history. In 1981, this IVF calf was born, and 86, these IVF protocols in 88. So this is a well-established technology done abroad many years back. Now, use of IVF in cattle. What are the main usefulness? If you have an animal with a dysfunctional reproductive anatomy, no problem. What we need is only ovaries to take out the oocytes. There is no, those animals which they don't respond to the gonadotrophin, they can be used as a donor. And you can use sick or injured females. Young heifers, let me tell you, it could be a 6-month-old heifer and you can still produce calves out of her, not directly from her, but you can take out the oocytes. As long as the hand goes into the rectum, you can take out the oocytes from such 6-month pre-pubertal heifers, and you can produce the calves out of her by making IVF embryos and implanting into other recipients. So she will take another 2 years to 3 years to produce her own calf through her womb, but already she will have her calves lined up before her calf comes out of her own womb. Even these IVF calves are her only, but they are born through the surrogate mothers. So you can do repeated oocyte collections in IVF. And another interesting thing, even if a donor is a pregnant donor, you can still take out the oocytes from the ovaries almost for 3 months of pregnancy. Sex-sorted semen is very commonly used into our IVF program. Some of the semen is very, very expensive. I don't know whether you have heard there are semen doses which are costing in thousands of dollars a straw. Such sort of a very expensive semen can be utilized on much more number of oocytes. Some definitions which I'll be using it into my presentation is that in vitro is outside, oocytes is unfertilized egg, ovulation, I think almost all of you know. Ovum. Ovum is again Latin word for the egg. Oocyte is an unfertilized egg, and ovum is also unfertilized egg, but same thing, ovum, oocyte. Oocyte is the immature one which we are taking out. Recipient is the one which receives the embryo. Capacitation, as some of you may be knowing, is a process where the insemination is done that sperm need to get capable to do the fertilization. A donor is an animal from which we take out the oocytes and produce the embryos. Embryo is a fertilized egg, and fetus and follicle are the structures on the ovary which are punctured and from which you take out the oocytes. See, these are the different terminologies in IVF. OPU is you pick the oocytes, aspirate the oocyte with ultrasound guided aspiration. I'll show you. And the next step is IVM, IVF, IVC, IVP, COC. So you will see these steps one by one. This is the state of the art of the laboratory of JK Trust, located near Pune, about 70 kilometers away from Pune. And now this is, you can see at a glance view of the lab. I definitely have worked into this lab. It's a world-class lab with the best possible equipments from all over the world. And even the Secretary Animal Husbandry, Government of India, Mr. Atul Chaturvedi, he had visited our lab and he had all the appreciation for this lab. Moreover, our laboratory is also recognized for giving a training, 1 month intensive training into the IVF also. So these are the different equipments. This is an inverted microscope. This is an embryo freezing machine. Freezing of embryos is a very different thing as compared to the freezing of bull semen. When we are freezing semen in one straw, you are putting almost about 20 million sperms, 20 million. Whereas when you are freezing embryo, please keep it in mind that in one straw, there is only one embryo. So how critical that whole process is, you should know it. When we are doing artificial insemination with the 20 million sperms in a straw, after thawing, immediately 50% of them, they die. And only one lucky sperm reaches to the ovum and fertilizes and resulting into an embryo. But whereas when we are doing embryo transfer, it's one single embryo which you are transferring it. So you need a good skills and a good practice. These are the benchtop incubators used in human beings also, where after fertilization, the embryos are kept here for 6 days to develop before they can be used for transfer. Now, these are the different lab equipments, and this is the chute and all that. Now, let me explain the reproductive tract of a cow. See, this is how the one's view of the reproductive tract, wherein you can see these two ovaries are there, and then the fallopian tube and the uterus and this vaginal fold, and this is how the thing is there. Now, in case of MOET, you are now, in case of MOET, once from the ovaries, the eggs are being released, you do insemination, they get fertilized here, and then these embryos are somewhere here into the tip of uterine horn, and through a catheter which is being put directly into the uterus. First, you have to put into the left horn and then right horn, and then you flush them out. That is collection of embryos, flushing of embryos for MOET, as I told you, where embryos are made inside the uterus. When we do IVF, we don't put anything inside the uterus. Through the vaginal fold here, we just pierce the needle here and just directly go to the ovary. See, from the needle, you just go to take the needle and you see everything through the ultrasound. There is a ultrasound guided aspiration you call, and on the ovary you will find the follicles, a lot of follicles. In our indigenous cattle, you don't need stimulation. So you attack one by one follicle and then you rupture them and you collect those. One oocyte will be there in one follicle, and through the suction, through a special tubing, a closed system, you collect them. So this is another view, same thing. Now, again here, we are again showing the lateral, another view of the whole thing, uterus. Now, this is what I was telling. See, this portion, this is what you are seeing, is known as the OPU handle. OPU handle. And in one hand is in the rectum, and I'm touching I'm holding the ovary. And the another one is that this ultrasound probe is being put through the vagina, and you are just touching, going to the ovary. You are not touching the uterus. You are going to the ovary, and there is a needle which is which is attached at the end. So it really, your one eye has to be on the ultrasound machine, and then you have to make sure that you don't prick your fingers rather than pricking the follicles, because they are very, very sharp echogenic needles are there. And particularly during a time of training, you normally prick your fingers and then you go through that horrible pain almost for 10 days. I'm not scaring all of you, any one of you, but that's how one has to learn. Unless and until you have one or two pricks, you will not understand how interesting is this whole technology. And through this suction, this is connected to a suction, and we put a media, what we call OPU recovery media, through which those oocytes are being aspirated. See, anytime you take a picture of an ovary, you will find a different type of follicles, primary, secondary, tertiary, and there can be corpus luteum, so different structure. So all these follicles which are there, each follicle will have one oocyte in that. See, normal reproduction, when a follicle grows, we call it as a mature Graafian follicle, as you all know, and then it bursts out and it will release one egg and it will get fertilized. And then at that place, corpus luteum will develop. But when we are doing this OPU, you have a follicles of different sizes, 3 to 4 mm to even up to 10 mm. So you take the needle inside the follicle, inside the follicle, and in ultrasound images, you will see them black color. So once the needle goes in, then you press with your suction needle, suction paddle, and it will puncture that follicle, and it will suck the oocyte from that follicle. That's very interesting. So the recovery of the oocytes, it will vary from animal to animal, but on an average, you get about 10 to 15 oocytes without any problem. But we have even touched more than 100 oocytes from a cow. So those are exceptions. So these are the follicular waves occurring at different parts of the estrous cycle. Now, I don't want to go into the stimulation protocol. I can share this presentation. Those who are interested, they can have a look at it subsequently. But in our indigenous animal, you take indigenous breed, you don't need to do any stimulation. But when you want to work on Holstein Friesian or Jersey or crossbreeds, you do need to stimulate them. You have to stimulate them with the follicle stimulating hormone. And in case of buffaloes also, we have done it. And in case of buffaloes also, you need a stimulation. Now, this is the protocol for the buffalo. Now, these are the different equipments which are being there. You know, this is a suction pump. And sorry, this is a heat block. One thing we have to keep it in mind, all these oocytes, they always love to be outside in a body temperature, 37 degrees centigrade. So everything, I mean, you may perspire, but the oocytes, you should not do them, keep them into too cold or like that. So these are heat blocks wherein you keep media also at 37 degrees centigrade, and whole operation is done at 37. This is the needle holder I was telling you. You can see that. And at the end, this is a probe which gets connected to this ultrasound machine. This is a portable ultrasound machine. And this is a tube in which the tubing goes through it, a steel tube. And at the end, you can see this small white portion. That's where the needle is connected. And this is all preparations you have to do it for the ultrasound. Now, this is the OPU needle holder, as I told you. On that, you have to put another, this plastic sort of a thing. Is nothing but it works like a condom so that it doesn't, break. And you attach you put the lube over here, and then you insert the needle. This is what you have to prepare it for inserting into the vagina, and then you go directly through the cervical fold. Now, I hope you recognize me. This is me only. And this is a chute wherein we are collecting these oocytes, one eye on the ultrasound machine, and then one hand into the rectum, and then this transvaginal probe into the vagina. So all you needed to have a good synchronization of everything. Now, this is the picture of a ovary, ultrasound picture, and everything you find, these are the small different type of follicles. Each follicle will have inside a ovum in it. I hope this is working. The video, you can see this needle coming from the top. Then you probably, I hope you see this, and it goes into the follicle. The moment it is sucked, the follicle, it becomes white. From the black, it is along with the fluid. See, continuously so many follicles are there on this ovary, and this needle is coming from the top. This is the image, ultrasound image of an ovary. We do left ovary, and we repeat it on the right ovary, the same process. So this is the way the oocytes are being collected. And now the next step is, this is what a graphene follicle looks like, in which we are putting the follicular fluid is there. And this is how we are aspirating a ovum. Now, what are the different things which will affect these OPU process? What is the age of that cow, operator skill, needle gauge, how much pressure you use it. Now, once you collect that, see, this is the tubing in which this is being collected from the ovaries, both ovary, right ovary, left ovary. You will get some blood tinge, but not to worry. And there is a small filter on the top. So you have to filter that from that tube which is collected, another tube. So ultimately, you have to search those oocytes. So you will have these oocytes collected from that, and this is how this picture of the oocyte looks under the microscope. These are the immature oocytes. See, this is, each one is a one oocyte, immature egg or oocyte, surrounded by cells known as cumulus cells. More the number of the cells around the egg oocyte, this is a good oocyte, A grade oocyte. Then you have to have, sorry, B grade. Oh, I got. So a B grade oocytes will have a lesser number of the cumulus cells. C grade will have a still lesser, and D and E, no, is almost, D is almost mature oocytes. We don't want mature oocytes. We want to mature them into the lab for 24 hours. Now they are in a particular specific media. See, this is what when we collected it. After 22 to 24 hours post maturation, these, they look like this. So you have to put into a in vitro maturation media. All these medias we have to import from other countries. And then this media, you are keeping it into the benchtop incubator, which I showed you. In that benchtop incubator, you are getting a gas supply of 3 different gases: oxygen, nitrogen, and carbon dioxide, the combination. And then these are the micro Q incubators. See, if I am and say, you invite me tomorrow in Srinagar, and I come and aspirate some of your good cows, and then I'll take those oocytes into a small vial and put into this incubator. And this is maintained at 37.5 degrees centigrade. And you just close it. And I take a flight from Srinagar to Pune. I bring these oocytes, produce the embryos into the lab. After 8 days, again, these embryos, I bring them back and put in your recipients of your state in your area. That's how these incubators, you are transporting these oocytes or the embryos by flight. So it should reach within 24 hours to your lab. Many times I tell the airline people, I have got a lot of passengers without any air ticket. They are traveling with me. So then now next step comes is the IVF, which is very critical, that you need to study the semen evaluation, a detailed examination of the semen to be used. Normally we have been using sex semen, so we get 90% of the female only and only 10% male. So it's choice, but extensive examination of the semen is to be done. We have to decide as to how many sperms are required to be put along with how many oocytes. So there is a lot of calculations involved into it. So I don't want to get into these extreme details. So ultimately you work out as to how many sperms. You do complete evaluation, semen evaluation study part, and then you decide how many sperms to be put. And this is how you do the centrifugation of the semen. You take out a straw, and that goes through all the process. Ultimately, you see this is the washed semen. At the end of the tube, you will find a small, this white pellet. This is the concentrated live sperms. All the dead sperms are here, so it is being thrown. And from this, you take out a number of quantity required, which is in microliter, and then you work out the quantity you have to put with the oocyte. Now, this is the process known as IVF. Under the microscope, you put this semen into this along with the oocytes, and this is a step second day done, known as in vitro fertilization. And then after that, these oocytes are kept inside. Once you put the semen, then you keep them again into the incubator. See how interesting it is. These are the 2 big oocytes, 3 oocytes. Now we have introduced sperms into that. And only, see, everybody is trying to rush and get into it. And as all of you know, only there is no entry after 1 sperm. Once 1 sperm gets into it, there is this chemotexis is there, and no more entry. But you will see it's a small video how the sperms are getting in and trying to get into the oocyte. You can see how the sperms are moving it, and they will try to get into the oocyte. So this is a small video. Now, you can use the frozen sex semen, as I told you, which has a lot of advantages. Now, I don't want to get, probably the sex semen is another technology which is being introduced on a very large scale, going to be introduced in India by National Dairy Development Board and the Animal Husbandry, wherein you sort out the X and the Y bearing chromosomes, and due to the DNA weight of these chromosomes, they get sorted out. There are two agencies in the world who are doing the work on this. Now, this third step, on second day, you have done IVF, and then those dishes are kept under that benchtop incubator, which I showed you. And then this is the third day, you take them out, these dishes, and this is how they look like. The oocyte you have seen, everything is outside, is being cleaned. On third day, you suck them out, and then again leave it. So any cumulus cells around that is being cleaned. Now we call them as a potential zygote or presumptive zygote. We are not sure whether they are going to develop into an embryo or not. Because say if you take 100 of this, not all 100 will turn out to be a good embryo. Only 35 to 40% will develop it, rest they will get arrested, they will not develop. Many reasons are there which is difficult to pinpoint. So these are the potential zygote on third day. You clean them nicely under the microscope and you keep them for another 6 days under that same benchtop incubator. Now, let's me come to this benchtop incubator. Very, very critical instrument, 38.5 degrees centigrade. Always electricity has to be on because you have got a live below this incubator. In those dishes, those embryos are there, and you are providing all the nutrients through the media which they would have got into the uterus. So it's nothing but an artificial uterus which you are creating into your lab and having all the sterility into the lab. Because if you have any problem anywhere in the infection, no embryo will develop it. So it is to be maintained at that. And there is a gas connection. There is a tri-gas of oxygen, nitrogen, and the carbon dioxide connected to that. So they will get media, they will get the nutrients, and they will also get gas. Now, this is a one cell, as you know, then it becomes a 2-cell embryo. This is when fertilization has occurred. Then it's not necessarily that it will develop into the embryo, 7-day embryo we want it. But multiplication goes like that only, from 2-cell to 4-cell, 8-cell, 16-cell, early morina. My sheep work, always you will see this 8-cell or 16-cell. You don't go further beyond that. Sheep or goat work, whatever we have done it. Now, these are the beautiful 7-day-old embryo. This is how in IVF we call early blastocyst. These are all 3 early blastocyst, full-grown blastocyst, and this is expanded blastocyst. This is on 8th day. So it's about to, then it will hatch. On 9th day, it will break the zona pellucida and the embryo will come out, and then it gets attached to the uterus on uterine wall on 14th day. So these are the beautiful IVF embryos. Now you should have recipients to transfer them. Recipient in which you want to transfer the embryo should also have come on the heat on the day on which we are doing OPU of our donor, so that the age of the embryo and then the recipient's reproductive tract is in the similar condition. Any one of you just think of whatever questions you want to ask, you can ask me later. Now, this is a bunch of embryos which are being collected from one single cow. This should be interesting. Look at this date down below. It is 22nd of October 2008, right? And time is 6 p.m. 9 seconds. Now, these are the human embryos. This is a live shot taken by putting an embryo, the camera into the uterus, and this is, I brought it from USA, how the human embryos, how the fertilization occurred and how they develop it. But you have to keep these have been identified, these are live embryos, and they have been identified with different alphabets. It's a few seconds video, and every second you will see the clock also here, how the clock is moving, date is changing. We are on 22nd of October. I think it goes and over at 26th of October, and then timing also will change. So I'm just starting this. I have to watch it very carefully, how they are developing it, and then ultimately they will hatch out, and then the embryo, it will come out from the zona pellucida. So see this. Oh, sorry. Oh, whatever. You can look at the clock. See, every second it is moving. So now it's gone to 23rd October. Look at the clock and look at the embryo's division also. This is how the cells are increasing. Now it has really advanced. This one very well advanced. This is how the multiplication, 8 cell, 16 cell, they're about to now get expanded. It's a beautiful, you can see it is 24th October. You can look at this third day, how this is happening. It's like a bunch of grapes. That is how it looks like. Now we are on 25th of October, third day, after the insemination, after the IVF. And every second the photographs are caught. The A1, this A is really developing very fast. Now you see how it is. This one also. All of them, they are gradually, and they will start hatching also. This 26th October, we already come on 4th day. You will see the hatching now, how it is coming. It's a very, very highly expanded now. See the zona pellucida, it becomes very thin. See, this is hatch now. The H has hatched. It has come out of this cover. So each one of them, see, this is hatch now. So you are on 20, this is over. The film is over. We are on this 27th of October. After 5 days, this is how the picture. So to describe you in short, these are the donor cows, the whole flow diagram. You do OPU, you collect the oocytes, immature, you fertilize them, you put into the benchtop incubator, culture them. These are the embryos. You transfer them into the recipient. You have an extra embryos, you can freeze them with a freezing machine. The 2 different methods of freezing are also there, slow freezing and vitrification. The vitrification is the one which is being used in human beings. Something is wrong with my sound today. I don't know, little bad throat. Otherwise, my voice is very strong that way. So these are the different logbooks. You have to use it to make sure that everything is perfect. This is a very, very interesting equipment known as a lab mix incubator. This is normally we use it. It has got a 38.5 degree temperature. The other incubator don't have a gas supply, but it has got inbuilt gas supply, tri-gas supply. So in this, we carry the embryos, we carry the oocytes across the country. We have done work right from north, right up to Kerala also, except we could not come to Kashmir, but we will definitely love to do that. See, these are the embryos. This is another incubator wherein you can see these yellow plugs here. And the embryo straws are kept here. We have an IVF mobile lab also. The whole process is showing about the transfer. We have a fleet of IVF mobile labs, one of its kind in the country. And even they're really is a very, very good van, fully equipped with all the equipments. Now, coming back, again, I want to stress upon the difference between multiple ovulation embryo transfer and ovum pickup. As I told you, we have to do flushing on day 6th and 7th. And here in case of ovum pickup, as I explained the whole process. Now, what is the ultimate? Through the MOET, embryo transfer, you can have, say, about 6 embryos per flush, and 2 months apart, you can do 6 times. So you can have 9 calves maximum. And that too also, you can't repeat on this cow for many years. But OPU, you can imagine, you have 4 embryo per OPU. Every 15 days you can do. You have 38 calves in 1 year. We have touched to almost 100 calves. That is different story, but that can be also done. So this is differences between the, now I'm almost coming close to my presentation, that we have worked in different breeds of cows, Gir, Sahiwal, Tharparkar, all different breeds, in exotic breeds and also in buffaloes. Recently, we did work in Gujarat on a breed known as a Bunny buffalo, which also we have got calves out of that. See, at our farm, we have got these elite donor cows. We have got, these are very high milk yielding donor cows. This is a Gir, these are the Sahiwal. Then these are the Jersey. Very interesting. All these donors Jersey, are born out of imported Jersey embryos from United States. These were all, they have all come as embryos into liquid nitrogen, and we transferred them. We got the calves born, and now they are almost about 2-year-old, and they have become donors for production of the embryos. Similar way, this is born out of HF embryo imported from Cemex, Canada. So JK tested a lot of credits, their first in IVF male calf, and they had worked in almost 34 farms across the country. So they have a lot of achievements are there. Now this is our mobile lab. We have done at different places. This is in Maharashtra. You can see these are the different calves born into Maharashtra state. You have a HF crossbred cow, then you will have a Gir calf. You can make any combination, any combination. This is a Kankraj breed of cow with a Gir calf. She is the foster mother, and this is the Gir calf. These are the identical twins born out of IVF. So this is in Punjab. We have done a lot of work in repeat breeder cows. You can use the donors. Donor is no problem. Even if they are repeat breeder, we can still produce the calves. These are the champion cows which were not getting pregnant for 2 years. And we produce embryos and we produce calves by using the recipient cows. So this is the beauty. I think this is in Tamil Nadu, Andhra Pradesh. We did it, as I was telling you, Andhra Pradesh Veterinary University. They have a project, and we did this with the Andhra Pradesh at Tirupati Veterinary University. And this is a Tharparkar cow with 3 of her calves. She is the donor, but this is their foster mothers. So this is, this is the praise. The another interesting cow, his name is Radha, and she had about 14 calves we produced from her by using the surrogate mother. But she is one of our champion cow known as Gauri. And now she has touched to almost 125 pregnancies. These are her embryos just in a span of 2 years. These are all her calves out of the 125 I'm talking about. The same cow's calf. She would have taken 125 years to produce that, but through this technology, in 1.5 years, they have produced so much. There's another lovely cow known as Samadhi, and we recovered, 103 oocytes from this cow, and produced 42 IVF embryos. That was the record in the country, highest record. And we have done IVF in buffaloes also, first time in the country. This buffalo, we have got beautiful embryos, and we got 3 calves out of this buffalo also. So this was also a unique work in the buffaloes also. So these are the buffalo IVF calves which are born. In one buffalo, we had twins. And now recently we did work with the bunny buffalo, which was highly appreciated by our Honorable Prime Minister also. This work is Mr. Atul Chaturvedi, who visited our lab, who is the current Secretary Animal Husbandry Government of India. And he had all the praise, and our farm is recognized for that. And this is my favorite picture of me with these lovely calves. Thank you very much. So that's the end of my presentation. Thank you.
Thanks a lot, sir. Thanks a lot, sir, for this detailed presentation, and very interesting. It led me to my young days when I was doing my FVSc in the same field. From Ranchi Veterinary College in 1991.
Okay.
So okay. I also did my post-graduation in vitro maturation and fertilization of goat oocytes. And they were at that time using the slaughterhouse material. So we're aspirating the oocytes from the ovaries, getting from the slaughterhouse, then maturing them.
Can you put your video on? I want to see you.
Sir, okay, I am here.
Just click on your video.
Sure, sir. So I am here.
Thank you. Yeah.
So it took me to those young days when I was a chap of 24 or something like that.
Sir, we are always young. I'm 71, but I say everybody I'm 17.
Then that was my MVC. During my period, I could have some, but that was not taken over. Then I just got absorbed in the animal husbandry department. You know that in the animal husbandry department we were concerned with some treatments and other things. That very late probably some 20 years now, I shifted to College of Veterinary Sciences.
Okay.
So this was really it. Just recollected my own days, those days at that time. My dear students, it was a very nice presentation. Before I will just open this for discussion. Let me have some clarifications from the speaker. That one, this your JK Trust, is it having some, I mean, the job opportunities for our veterinarians there? Do you just recruit the veterinarians there? One. Second, do you get do you offer these training programs to the veterinarians and you train our veterinarians there? And is it free or they will have to pay fee for that? Then you provide some certificates to them. uh, this is what is basically related to my students. If they want to have training for this embryo transfer, is it free or it is paid? Then secondly, is there opportunities to get them absorbed in your JK Trust or like that? Please.
I mean, there is opportunity. They should. I will share the ID of JK Trust. They should send it an email with their resume, and the training is there is some cost involved into that. So they can always send an email and get the details from the new CEO who has taken over this whole thing.
So they have the opportunities and they can get the training.
Yes.
Now, is this training for veterinarians or say any other non-vets also can have this training?
No, mainly, as per our veterinary council and all that, we have to have the OPU training, the training of taking out the oocytes and embryo transfer exclusively for the veterinarians. And on the laboratory training, it can be for the biotechnology, microbiologists. That's okay, no problem. For the MSc biotechnology or MSc microbiology, those who have worked into the laboratory, they can be very well trained into the lab techniques.
Okay. So, my dear participants over here, if you want to just have an interaction, I will love definitely to have the interaction with our this learned speaker of the day. Please go ahead. Please go ahead. Yes.
Good afternoon, sir.
Yes, you are audible.
Yes, sir. Thank you very much, sir, for your presentation, sir. It was a very nice presentation, sir, vis-a-vis your knowledge and hold over the subject, sir. But, sir, could you please elaborate as to how this technology is going to bring this technique into this entrepreneurship field, sir? I mean, say, sir, with respect to J&K, since you have yourself been associated with this state. So could you tell me, sir, how this is related to the entrepreneurship field, sir? I mean, say if someone wants to set a lab, sir, in Kashmir, how much it is going to cost him, and will it be fruitful for him to actually set the lab?
I mean, one can set a lab, and then, now recently some Government of India, Ministry of Animal Husbandry, I don't know the details, have come out with some infrastructure funds where you get a good funding from the government, and they are very keen in supporting such labs. So you should visit the Ministry of Animal Husbandry, Dairying and Fisheries, Government of India website, and I think you have all the details on that. Second thing is that technology is expensive, but if I think in your in your state, there will be I'm sure, with the climatical conditions, these exotic animals must be performing very, very well, in my opinion. These Holstein, Friesian, and Jersey, and if they have to be multiplied, it's a very good technique is there to multiply the very high milk yielding animals. So I'm sure you will be able to develop a clientele for that. It is expensive, but it is going to, in long run, you are producing a genetically very superior animal. So you have different choices. You can sell embryos, or you can sell pregnancies, or you can produce elite calves. There is another thing is that you have your own lab and you have some donors. See, everybody you may not have your own donors. So what we do, there are some farmers now, we have got some very good donor cows are there, very high milk yielding. So we give them a X amount, maybe 5,000 rupees, 10,000 rupees, and allow them, they should allow you to do the aspiration and take out the oocytes, and you pay him that honorarium, say 5,000 rupees. And he should be assured that nothing goes wrong with his cow. Even after doing 30, 40 aspirations of a cow, we are able to impregnate her anytime. You want to make her pregnant, no problem. But you have to develop the skills, you have to do the practice first before offering this. Then farmer once get in confidence with you, such farmers will come to you and you can take out the oocyte, produce the embryos, some such good cows using a good sex semen, and sell those embryos, or transfer those embryos and sell the pregnancies or sell the calves. Many options are there.
Sir, actually, I wanted to know you see, sir, let's assume, sir, if I want to set a lab and I have already, for example, let's assume, sir, I have set a lab of this embryo transfer technology, sir. Sir, I want to know, will it be a fruitful entrepreneurship for me, sir?
Yes,
How much
it will be.
it is, sir?
It will be
Sir,
fruitful. It will be.
keeping in view the average field conditions, sir, because at last I would have to transfer the embryo in the cows in the field, sir.
See, what
That
I suggest to you, you should be not make this as a only complete thinking of making complete income out of it. You should be give your services as a consultant, as a ART consultant. It could be in artificial insemination, it could be in embryo transfer. So it's a reproductive biology, reproductive biology expert. And for that, ultrasonography is very, very important. You must master the technique of ultrasonography. That will really help you a lot. There are people who are only doing ultrasonography and no need even for IVF and all that. And they are doing very well. There are experts like that.
Okay, sir. Thank you so much. Thank you so much for your time and answers.
Hello, sir. Hello, sir.
Yes,
Uh,
yes,
sir,
yes, yes.
sir, thank you for your
Dr.
excellent
Saeed Shahnaz
presentation.
is with us, sir. He is from the division of breeding and genetics. She's there, the associate professor, and likely to be the head of the division. So, Saeed Shahnaz, most welcome. Go ahead with your query, please.
Sir, sir, it was a very excellent presentation and we got to know many things out of it. But one thing is there, seen in humans also, there is one dreadful complication associated with this embryo transfer technology, that is OHSS, wherein yet there is ovarian hyperstimulation syndrome. And most of the females, I mean, from the human side also, they hesitate to go for this technique. See, I have seen some of the one of the very fatal cases of this, one of our neighbors, she, after 7 years of her marriage, she was luring for the child and she went for this thing. And she is no more because of this complication. What actually can we do to prevent it, and how we can prevent this hyperstimulation of this ovarian ovaries to produce more of the oocytes? And how can we get it in a fruitful direction so that we harvest most of the oocytes in a well, I mean, I missed that part. Maybe I got disconnected at the time when dose, or when the technique was being asked. You were presenting. Is there some dosation with it that to avoid this?
Yeah. Let me tell you. See, first of all, Bos indicus and Bos taurus. Let's differentiate that. So all the animals coming under Bos indicus, which are our indigenous breeds of animals in India, they don't need stimulation at all. Number one, rule out that we don't need to do any stimulation. In case of when we are doing MOET, flushing, that time definitely you need to do a little stimulation to follicle stimulating hormone, FSH, which is a very excellent product. No problem with that. I have been using my whole life. And the dose, you don't need to do overstimulation, You have to have the dose perfectly. But in case of IVF, when you are working with the Bos indicus animals, no stimulation is required with any of these our indigenous breed. Now, when we are talking about HF and Jersey, whole world is using it. I've been seeing it, and they are stimulating it and they are collecting it. But that stimulation also, it has to be a proper dose. And in case of it may happen once in a while, a case like the what you have told just now in human being. Even I share with you, even epidural anesthesia, epidural anesthesia also, there are chances that you may lose some animal once in a thousand animals. You do epidural anesthesia, animal will sit down and will never get up again, will die. So such cases do happen, but that cannot be taken as they are very exceptional things. So that doesn't come in the way of doing this work. Thank you.
Okay, sir. Thank you, sir. Is there any
Sir.
correlation between this IVF embryo transfer MOET with this setting up labs for this indigenous animals, and we have to have different criteria for indigenous animals for establishing a nucleus, rather wherein we do correlate MOET with like we have juvenile MOET, we have adult MOET, wherein we correlate the technique with setting up a nucleus for further conservation or propagation to the breeders. Is there
No,
any species?
No. You can have one lab, and then you can do everything in that. There is no need of a separate lab or anything. And very well, these labs can be established. I'm telling you, when I started learning, you don't need, every cycle, there will be one egg will be shed by the cow. Am I right? At the time of ovulation. So you try to aspirate that one, try to flush out that one egg from the uterus. And if you can recover out that one egg, that means you are really doing good. You don't need any expenditure, nothing. You can use just a normal saline and do the flushing practice. This is as far as the MOET is concerned. Now, in case of IVF, you take some cull animals are there and go on practicing it. Count the number of the follicles and see how many oocytes you get. What sort of a recovery percentage is there? And if you don't want to culture them into embryo and all that, you can just use simply normal saline just to know as to how many oocytes are collected. So training can be given in a different ways without any much of the expenditure in that.
Sir,
But
excuse,
ultrasound.
sir, one more question is there.
Hmm.
Most of the times there is a problem of tubal pregnancies with this ARTs. Is it, sir? Is it in animals also? We have, since we are not knowing it.
No.
Because of this ARTs, we have encountered.
No. We don't have that problem, except I can tell you there is one problem which occurs with the IVF is that there is a large calf syndrome. You get a larger calves in case of, particularly if they are males born, no?
Yes, sir.
Large
Yes,
calf
sir.
syndrome, they call it. They become, and then in case of IVF, you sometimes get more abortions and the stillbirth. That is another thing. And freezing of embryos in case of in vivo, you get a better results as compared to freezing of the IVF embryos. A lot of work is going on all over the world to improve the pregnancy rate with the IVF frozen embryos as well.
Okay, sir. Thank you very much.
We have with us Professor Mujib Fazli. He is basically this time in charge of cattle breeding farm in Kashmir there. And he has been associated with this embryo transfer technology for a pretty long time now. And he has been the man behind that success story of Noori, that first clone that was born there in our state. And he is very expert in that, you see, laparoscopic surgeries and then laparoscopic transfer of embryos. He's with us. Most welcome, sir, Professor Mujib Fazli. Please go ahead with your queries, sir.
Thank you, sir. Thank you, Professor Pampuri Saab. First of all, I will thank you for presenting me in a way which I am humble. I am not of that quality that you have told. Actually, sir, I was very much I'm thankful to you for this nice presentation. I actually started a bit late, so you might have already given those queries which came to my mind. You might have already explained them, but for me, kindly repeat them if you have already told. And regarding this, myself, I am actually a surgeon, veterinary surgeon, and I am still associated with that embryo transfer laparoscopically, usually for small ruminants in sheep and goats. We have been doing it since last few years in Faculty of Veterinary Sciences, Srinagar. So, but regarding this topic, sir, you have nicely presented it, and rather I got elated that we do have such actual facilities within our country presently. Otherwise, we the old students used to simply to learn from books, etc., and not get actual exposure to the people who actually do these things with their own hands. So very nice presentation it was. I have just three things for learning purpose. I have number one is the name of your that organization is JK something.
Yeah.
So we are from JK. So is it having any relation to the JK? Because
No, no, it's
I
very,
got
very,
affiliated. Huh?
it's very, very interesting. No, that's why we our JK always wanted to work in JK, but we didn't get an opportunity. JK is the initial of the founder of this organization. Jugilal Kamlapat is their name and the father's name. So this is how it's known as JK organization. Our principal organization is JK organization. You must have heard about JK Tire. You must have
Yeah,
heard
yeah,
about JK Paper and
yeah,
like
yeah.
that. And Raymond Shootings, which everybody knows, is a part of JK organization.
Okay, okay, okay.
So the JK stands with a hammer, the symbol of hammer. So that's the organization. Yeah. And as you rightly said, you are a surgeon. You know, I wish I would have been when I passed out in 1972, my graduation. And that time when I was working with the sheep, it was all laparotomy. And I must have done, like you have done thousands of laparoscopy. That time there was no other choice, but I must have done around 10,000 laparotomy. We used
Okay,
to hang the sheep upside down in a laparotomy cradle. It was and then tie the legs on the top, hind legs, and then I used to sit on a stool, open up, take out the uterus with a finger, and then do a surgical transfer. I used to do that, and then second fellow will do muscle suture, and third fellow, my lab boy, will do the skin suture. Like that, we used to do at least about 200, 300 transfers every day, every
Oh,
day.
oh, 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 oh.
So
Yeah, that
that
had
was the
been
volume
hectic.
of work we had done it long
Uh,
back. That was from 1975 till 1983. All those years, right from morning till evening, that was our work.
that's great to
Yeah,
learn that actually those hard tasks, they give you the experience. That's why the way you are speaking and you are presenting, that indicates that you have worked hard throughout your life uh,
sir.
and come
Every day,
level of,
day in and out, I only think of embryos, cows,
okay,
and
okay.
livestock.
Yeah, yeah. Uh,
My mobile phone is full of pictures of embryos and cows only.
okay. Now, although JK, as you have explained it, it does not mean Jammu and Kashmir, it means
Yeah,
something else there.
yeah,
But
yeah.
let's be that optimistic. We have some affiliation to the word JK, so we'll be remembering this name. Yeah,
Good.
and yeah. But second query that I want to just highlight is that uh, it's very fine that we can now we have this lab with you, or there are people who are producing embryos, who are preserving embryos, who are transferring embryos, etc. But the level in our field conditions is that our farmers have 1 or 2 cows, and even if they are yielding 5 liters, 6 liters of milk, we this is not the stage that where they will be accepting our this technology, where we tell them that okay, we'll transfer embryos and you will get a calf and that will produce 20 liters, 30 liters, etc., etc. Maybe in times to come, we have some organized farms where this type of technology will have high potential for application directly. But the problem that we are nowadays facing is that many of the youngsters educate earlier, it was this till now also, the livestock is reared mostly by illiterate or low income groups in throughout our valley at least.
Yeah.
So, but now since there is unemployment, now our educated youth have started coming in for entrepreneurship with MBA, with post graduations, etc., etc. And they and governments has also shifted their focus towards the livestock. And now there are various schemes with animal husbandry, sheep husbandry department, that they give them subsidies, etc., etc., which was not available earlier. So the government also wants to boost this livestock industry. So now those potential entrepreneurs daily visit our farm and interact with us. And most of them get convinced with the benefits that they may be getting out of these livestock related activities. But once we finish convincing them in our way or presenting, then finally they the last question at which blushes us most of the time is that, okay, sir, I'll now have a dairy farm, I'll have 10 cows, 15 cows, but where from shall I get the elite animals, even if it is costly? And we don't have any source of those animals to provide to them. They will not be waiting, they will not be getting the inferior quality animal, and then we'll transfer the embryos, and then they, after 9 months it will calve, and after 2 years that calf will come to the estrus or become cow. So they are not ready to wait for that. Is there any shortcut where we'll be able to provide them better quality females?
Sir, let me answer a few of your questions. Like, as you rightly said, see, there's no point in doing the work into the laboratory and just publish some 10, 20 publication. That doesn't really, I've been working with a private organization like Raymond. So our focus was always whenever we did it, this IVF work, we took it within 1 year, we took it to the doorstep of the farmer, and we have produced more than 500 calves at the doorstep of the farmer. There were a lot of reluctancy were there, a lot of illiterate people, they were not at all any good thing. No, it spreads, it takes a lot of time to spread, but anything bad thing happens, it spreads very fast.
Yeah.
So that way we have to really struggle it, and we could succeed it. Time will take. Another thing you asked me, shortcuts, sir, any time you go to any market in the country, I'm telling you, you will never get there's so much of cheating. I'm honestly telling you, you will not get a correct animal when you want to buy it. You have to have your own animal produced. It is better to wait, and rather than get a wrong animal and struggle with it and then get loaded with a bank loan and all that, and ultimately you lose the interest. So take your time and get the good animal, start with a good animal. It may take 3 years, but down the line, you will have a beautiful herd. And once at the farmer level, this experiment is successful and farmers go and visit it, I'm sure it will spread. Today, IVF was a dream in India. When we started in 2016, we really took up in a very big way. Today, there are 30 labs are there, and I'm so happy that I'm offering these honorary services to the whole country now. After putting from 1st of November, now I'll be visiting each and every lab and share my experience and try to see what sort of a bottlenecks they have got it and try to help them. That is my only purpose of taking this mentorship, which is something a new sort of a designation. I'm very happy with that. And I hope I can do something for the country.
Yes, sir, I am optimistic. I'm optimistic. The educated youth who are now coming towards this livestock industry, they will be establishing mini farms, 10, 12, 20 animals, and then hopefully we'll be able to convince them better than the lot that is nowadays available with 1 or 2 cows per household. So hopefully this technology will reach Kashmir also and our villages also. But
your weather conditions in Kashmir are so beautiful. Any animal into the plain, what they produce it, I'm sure they will produce wonderful over there. The record should be very, very good. I feel like that.
Yeah, that's true. Sir, one more, just be educated about one thing. I'm, as Professor Pampuri said, that I am nowadays in charge of the dairy farm of the university. So during, I have been there since last 1 year or so. And during this period, almost 30 cows calved. We have, it's a small farm. It's a small farm having hardly 100.
Which breed?
We have Jersey and HF, mostly Jersey, but HF are some. So 30 cows calved, and I was expecting the male-female ratio around 50%, maybe 55, 45 extra. But to my surprise, we got almost out of 30, 25 males instead of. Yeah, so again, I'm in a fix. So
No, you have
then
to
we
see
thought
what bull you have used it. You have to go into the history of that bull.
not
But
only one bull. We are getting cement from the Department of Animal Husbandry. Multiple bulls are there for Jersey, HF, HF. So there is no single bull is not used. Many
Okay.
cement from many bulls is used.
I think
Anyway, now
share coincidence probably, because it's normally at the end of the year, normally you get a 50% ratio.
exactly. But in my case, it has been like that. So, but now I thought of thinking over the problem. Then again, as you have already mentioned, saxid cement is the answer to this problem. But saxid cement is very costly, and it will not be possible for us to purchase for small number of animals. Maybe in bulk, if we people will ask. And is there any number one? Can you suggest some measures other than saxid cement?
No, I will
Number two,
suggest you, sir.
number two, regarding saxid cements, whether cheaply it could be available to us somewhere.
Sir, let me tell you something about sex semen. Sex semen is a very important topic taken up by the Ministry of Animal Husbandry, Government of India at all level. This morning I was having a lot of discussion. Now let me tell you, sex semen, the two companies which are in the world, they are doing the sex semen. One is known as sexing technology, which are very well known throughout the world. And another one is ABS, American Breeding Services. The difference between the technology used by both these agencies is that in sexing technology, they differentiate X and Y clearly, they separate it. In case of, and the Y sperms, when they are killed, they will out, they don't remain into the semen. Whereas in case of ABS, they kill the Y sperms, but then they remain inside the semen. So the conception rates of both the, I don't want to give my biased opinion, but the country is utilizing, farmers are using ABS sex semen also and ST sex semen. There are two agencies. Now let me tell you, a very big program is coming up by the Government of India. They have a plan to produce. This morning only I was looking at some, in 5 years time, they wanted to establish about each year 42,000 pregnancies with a sex semen. And the National Dairy Development Board has been an agency which has been given the job to get a bid. You can see this bid document on the NDDB site, and they are inviting the bid from these these are the only two players. And it is hope now that the cost of the present sex semen is around 900 to 1,000 rupees, but which will probably come down with this bid process to around 600 rupees. And government is going to planning to give about 50% subsidy on that. So those details, you can see it. But coming back to your question, you can get even a small quantity of semen also, no? From these you can get any number of doses you want. You want HF, Jersey, whichever breed you want, any breed you tell, and you will get about, for 800, 900 rupees, you will get the sex semen. Now, every state government, they are subsidizing it very heavily. For example, I'm in Maharashtra right now. The sex semen is made available to the farmer only for 81 rupees. Only 81 rupees. So cheaper, you know. Rest of the cost is taken up by the government. State government is taking care of that. Even though the producing company, the one which are these two companies which they are selling it, and they have got 7 sex sorted semen labs in the country. The ST technology has got at 3 places. One is in Uttarakhand, Uttaranchal Livestock Development Board. Another is in Bhopal, Madhya Pradesh Livestock Development Board. And third one is with the BIF near Pune, Urli Kanchan. Now, the ABS technology has got 4 places. They have, I think, in Uttar Pradesh, and they are in Maharashtra one. So like that, they have another 2. So there are 7 places where the sex semen is being produced of the different bulls, different breeds. So is in Kashmir, are you not using in the valley? Are you
not yet. Not yet. Maybe, hopefully, because of details provided by you, we hope that our Department of Animal Husbandry also maybe then doing something in this regard, and hopefully we'll also get.
I'm sure, sir, because you are.
I want to add over here, sir. I want to add over here. Basically, now in Valley of Kashmir, this dairy has been taken by a private sector. We have 2, 3 districts over here where comes this Pulwama, Shupian, and all that. I have personally visited many farms which do carry more than 15 animals, and that to pure these HF or jerseys, and they are. I have seen even one cow just a week or 2 weeks before producing 35 liters of milk in a day. So there are very good quality cows there, and I just inquired from them. They are getting this semen from Punjab, sexed semen from Punjab. But the problem with them is that again that repeat breeding, and they do not have the expertise with them to inseminate themselves. You see, I could see their farmers telling us, "Sir, you give us this a training for insemination. We will inseminate our cows ourselves. We will get the semen from Punjab. That's no problem with them." But the problem is that they do not have that expertise to inseminate the animals. For that, they are not interested to involve the animal husbandry people. A government these functionaries there, they don't want to involve them. They want that they should get this training of AI themselves, and they will get this semen, sexed semen from the Punjab, and they will do that job. From most of the people, I could sense that their insecurity with them. Otherwise, it is available. The interested private farmers, they do get it, and they manage it. I don't know how they manage it, but they do manage it. And we have very good farmers now coming up in the state. That's my personal experience. I have seen myself.
Sir, there is no problem. There is nothing to worry about getting the sex semen. As I told you, these are the 7 semen stations where from you did not go to Punjab or anybody. There is a list. You can go into the Google and ask which are the sex sorted semen laboratories in India. You'll find these 7, and you can get any breed you want it. You want 20, 30 doses. And another thing is that sex semen also giving 40% conception rate. There is no difference between the conception rate between the sex semen and the conventional semen, as long as the animals are good. If you want to try repeat breeder animal, many times our farmer know in our IVF work also, they thought that those cows which doesn't remain pregnant with the bull or AI, you just put embryo, they will remain pregnant. That's not a correct way. Okay. So you have to educate them, and sex semen you should preferably try with till the people get a confidence. They should use it on the heifers. If heifers, you will get a wonderful results on the sex semen. And then look at the history of the cow. Tell them they don't waste such an expensive semen on using on the repeat breeder cows which have not conceived it. So a good AI technician should be able to do this job without any problem. It has been used now almost, I think, 2 million inseminations with the sex semen is done in the country. 20 lakh inseminations are already done. So many thousands and thousands of calves are born.
Thank you very much. Thank you, sir.
I think this is the opportunity where our, the participants should look into, that they can have those forms where they can establish their own, pure breeds there, and they can just go for in its entrepreneurship and can propagate those calves, the female.
Sir, I request all the students here that they must visit the website of the National Dairy Development Board, study that website in detail. Then another thing is that they should visit the Ministry of Animal Husbandry website. Third application, I don't know how many of you use. You should always look at there is a small application, ResearchGate. I'm sure some of you may be using on your mobile. Use that ResearchGate application to know what's happening in the world, to keep yourself updated on the various things.
Thank you. So nice of you, sir. Sir, I think we have taken much of your time. See, there has been a very good message from yourself. I could sense that when you just told us that at the age of 70, even at 70, you are doing yourself the embryo transfer. So age is no bar there. So my dear students,
I'm not 71. I'm 17. Ulta, vice versa. 1 second.
Correct, sir. So I want that our students to be 16 only, not 17. So it's very, very interesting and very informative and very, I can say, inspiring for our students also. They need to have that passion, they need to have that dedication towards this profession. Before we take leave from our speaker, it's my moral duty just to thank our speaker, and for that I humbly just request our Hamdani, Shabir Hamdani, Dr. Shabir Hamdani is there. Shabir Hamdani, you may please unmute yourself and go with the vote of thanks, please. If you are there, Shabir Hamdani. I could see here, Shabir Hamdani, can you unmute yourself? Dr. Rawu Puttu.
Yes, sir. Yes, sir.
You are there, please,
Yes, sir,
please.
yes,
You see, this is our moral duty, and you are associated with this program. And
Yes.
he is basically the organizing this program, Dr. Rauf Ahmed Puttu. So let you propose the vote of thanks for our speaker of the day, please.
Thank you, sir. Thank you, sir. On behalf of organizing committee, I thank our eminent speaker for sparing his time and valuable suggestions and for encouraging us. I also thanks my faculty members for interacting with the eminent speakers and also the participants. Thank you, sir. Over to you, sir.
My dear. So we are looking forward more cooperation from our speaker of the day. We'll see that this embryo transfer technology, hopefully I will talk to the functionaries at the animal husbandry department and our department, also a reproduction department. We will see that embryo transfer technology lab is established there somewhere in the department or there in our university. And for that, we wish that you will support us wherever we need your support there. Thanks a lot, sir.
Yeah, I will definitely. You please tell your vice chancellor or dean, whatever, and send a project proposal to the ministry and request them. And I'm sure you will be able to under RGM, Rashtriya
Gokul Mission. Yes, sir.
Under
Yes, sir.
RGM, you should be able to get the funding like the other universities have got it. Why not? You should also try it with this.
Yes, sure, sir. Sure, sir. That's the message we have got over here, and hopefully we will do something in that direction. Thanks a lot, sir. Thanks a
And
lot.
I really enjoyed talking to a majority of you. And then one thing which I wanted to tell you is that during the lockdown period, to spread this technology of IVF, actually I could have done in your language also, but I didn't do it. But we did it in almost in 12 languages. It is there on the YouTube channel,
Okay.
IVF in cattle and buffalo, which is there in the regional 12 languages of the country. For spreading this message, I started with Hindi and then English and then all southern language, all northern language. So those who are interested to hear that, they are most welcome.
Please, you can share that video, sir, with us. That
No, you just put YouTube. You put JK Trust YouTube, you will have
Okay.
everything.
Okay.
Yes,
Okay.
JK is now, no need to forget that name.
Oh, no, we cannot forget JK. We cannot forget.
So
That's
JK
there.
goes,
So
JK goes very well. As I told you,
yeah.
I had visited Valley many times. Once even, I think I had come as a member of the planning commission also to Srinagar. And once there was some reproduction conference also, I remember. So I had come many times.
So now we will see you over here. That will be our honor to host you over here. Anytime you are there in Srinagar, please.
Yes,
be with us.
Just to add quickly, even I was invited by the Defense Research Organization. I went to Ladakh, and
Yes.
the field research laboratory, I did a embryo transfer in yak animals also.
Okay, good. So hopefully we will receive you there in Valley soon in the next year, inshallah.
Yeah.
Hopefully.
Thank you very much. I wish all the students all the best. Thank you very much. Thanks
a lot, sir. Thanks a lot.
Thank you. Bye.
Bye. Just sending the meeting over here.